Benthic macroinvertebrates are animals without a backbone. They're visible to the naked eye and live on river bottoms. Because of their sensitivity to physical and chemical changes in their habitat, they are excellent indicators of the overall health of the river. Among this group are insect larvae, crustaceans, and mollusks. Collecting macroinvertebrates requires many different pieces of equipment. Take the time to familiarize yourself with this equipment before going into the field. There are two methods for sampling macroinvertebrates. The physical characteristics of your site will determine which method you use, either a method for fast currents or one for slow currents. A fast-flowing river is characterized by a water flow with turbulence on the surface and a large-scale substrate made up generally of boulders, stones, and gravel. If your site matches this description, you should use the method for rocky riverbeds. On the other hand, a slow-flowing river is characterized by a calm surface and often has aquatic plants. The riverbed is generally made up of fine, silty, or sandy substrates. If your site matches this description, you should use the method for muddy riverbeds. Whichever method you use, remember that sampling macroinvertebrates is always done from downstream to upstream in order to avoid stepping on and disturbing the habitat to be sampled. Only the D-net can be used to sample muddy or sandy riverbeds. Start by identifying the habitats present on the site. For example, marshy shorelines with a large number of emergent aquatic plants. Submerged rocks or tree trunks. Beds of completely submerged aquatic plants. Silt, sand, or gravel lacking any vegetation. You should take a total of 20 samples distributed around the different habitats. The most productive habitats, such as marshy shorelines, aquatic plant beds, and tree trunks, should be sampled more frequently than the less productive habitats, such as silt, sand, and gravel. So let's say you find all of these habitats on your site. You might take 2 samples in the sand and distribute the remaining 18 by taking 6 samples in each of the 3 other habitats. The number of samples you take in each type of habitat should be recorded on the field chart. We will now go over the sampling procedures for each type of habitat. Note that each sample should last 30 seconds. We recommend emptying the net after each sample. For marshy shorelines, the person holding the net rubs the stems and roots of aquatic plants with the bottom of the net, while gradually moving the net upward. During this time, the other team members can hold the bucket, assist the net holder, keep track of time, or keep track of the number of samples taken. At sites with tree trunks, one person holds the net beneath the tree trunk being sampled, while another person brushes a 30-centimeter square area on the side of the trunk. Collect all the organisms, bits of bark, twigs, and leaves that are dislodged. In aquatic plant beds, the person holding the net vigorously scrapes the riverbed and the submerged plants while gradually moving the net upward. In habitats with silty, sandy, or gravel riverbeds, the person holding the net places it on the riverbed and pushes it upstream for 30 centimeters at a depth of about 5 centimeters, while stirring up the surface of the silt, sand, or gravel. To avoid collecting too much silt in the net, place it horizontally above the water, making sure the metal rim of the net is out of the water, and gently shake it to let the fine bits of mud fall through. Since sand, silt, and clay can make your sample very turbid, it is highly recommended to thoroughly rinse the sample by keeping the net in the water or by placing it in a wire mesh bucket. It is advisable to empty your D-net as often as possible, ideally after each sample, because the force of the current and the substrate could damage the macroinvertebrates trapped in the net. To empty your net, just turn it inside out over your tub or wire mesh bucket. Most of the net's contents will fall out. You can do a thorough cleaning after the last sample. To make sure you don't lose anything that might be left in the net, take care to put the net back in its original position after you empty it. After the last sample, clean the net thoroughly by using tweezers to remove any macroinvertebrates that might have become trapped in the mesh. Finally, rinse and dry the net properly for the next time it is used. If you collect your sample in a plastic tub, make sure you don't overfill it with water when rinsing or cleaning the net. Your tub should not have more than 2 centimeters of water in it. A wire mesh bucket is a very useful sampling tool. It allows samples to be thoroughly rinsed and cleaned before they're placed in a pot or tub for sorting and identification. A sample that is free of silt and clay and large debris makes the next steps that much easier. The goal of sorting the macroinvertebrates is to group similar organisms in your sample together. To do the sorting, use a pair of tweezers or a pipette with the end cut off. Sorting occurs in 2 stages. The initial rough sorting lasts 10 minutes. Then the subsampling lasts 5 minutes. Respecting these time limits is extremely important because they are part of Adopt a River's scientific protocol. If at all possible, we recommend that you do your sorting on a table or some other raised surface so that you'll be more comfortable. The goal of the initial rough sorting is to highlight as many different macroinvertebrates in your sample as possible. Use ice cube trays to group similar organisms. Be aware that certain macroinvertebrates are very small, and they can even be almost transparent. Keep your eyes open and study your sample very carefully so you don't miss any. The goal of the sub-sorting is also to highlight the sample's diversity as much as possible, but this time concentrating on a smaller sub-sample. Use a ladle to take subsamples that you then place in smaller containers. As a team, continue to remove all the different macroinvertebrates that you find for a period of 5 minutes. Even if there are macroinvertebrates remaining in your samples, make sure you stop sampling in order to respect the scientific protocol. When the time has expired, place your sample back in the river and rinse your containers with clean water. If you plan to do identification in class or in the lab, transfer the contents of each compartment of your ice cube trays into watertight containers. Your sample can be kept for a few days in a refrigerator. However, we advise you to do the identification on the same day. Otherwise, predators within the container will compromise your result. Also, dead macroinvertebrates may be harder to identify. Once you have finished sorting, it's time to count and identify the macroinvertebrates you have collected. To do this, use the visual identification key to freshwater benthic macroinvertebrates poster format, along with the macroinvertebrate identification key that can be found in the program guide. For each ice cube tray compartment, identify the order to which the macroinvertebrates belong. If a specimen happens to be missorted, place it in the proper compartment of the ice cube tray. Some macroinvertebrates can move quickly and may change compartments or even try to escape from the ice cube tray. Be alert. For each order, examine your sample and determine the number of kinds it has. You are now ready to complete the Counting Macroinvertebrates field chart. Simply count the number of macroinvertebrates in each order and the number of kinds. But be aware that for certain orders, the field chart lists specific kinds that you must identify and count separately. For example, for the order Diptera, the Tipulidae are recorded separately. So it is important that you don't recount them when counting the Diptera as a whole. Then, all that remains for you to do is enter these data into your internet report to obtain the index of the river's biological health for your sampling site or sites.